Okay, so hi everyone. So thank you very much for joining the section with the SCSE Club today. So thank you very much for spending your time to join the section today. And today I will have the show you about the application of the SpatialX in terms of like study different human diseases, including today I'm gonna focus on the inflammatory bowel disease or IBD at single cell level. And this is we're gonna use the single cell transcriptomic data. So here. So for everyone to be on the same page, so I give some information about the disease we're gonna focus today. So in terms of inflammatory bowel disease, which is the group of the chronic disorder that marked by the ongoing inflammation in the GI tract, and then patients usually experience with lots of like abdominal pain, diarrhea, rectal bleeding, and also fracture. And there are two main primary types of the IBD, which are the Crohn disease and colitis, and then depend on different area of the intestines. For example, here the Crohn disease can affect any segment of the GI tract, and it often extend to a deeper layer on the bowel wall. But the ulcerative colitis is more confined to the colon and the rectum area with the produce of the continuous inflammation of the mucosal lining. And one of the challenges of treating IBD is that there are wide variety in disease behavior, treatment response, and the progression among patients. So understanding more about the disease and also any heterogeneity between patient and patient are more important to develop a more effective treatment. And for the data set that I'm gonna use today is for confirm a published study from Trigo and college from Salas group. So this is a very nice study when they collect the sample from healthy control Crohn's disease and use ulcerative colitis and do both like single cell and also spatial transcriptomic. And this is their experimental design. So the group, the lab collected sample from human from the healthy control, which is SC here, the Crohn disease CD, and ulcerative colitis, which is UC here. And after collected the sample, they do both approaches. The first one is single cell RNA-seq to able to analyze the data at the single cell level and also get enough cell number and also get enough the gene to have the better cell type notation. And from that, they can do use that information to help to annotate cell in the spatial data and also for further other analysis as well. And the second approach that also used here is the spatial transcriptomic using the cosmic SMI. And after they do all of these two approach, they will transfer the label from the cell type notation from the single cell to the spatial transcriptomic data from the cosmic data. And they can assign the cell and then they can do further analysis about cells on the sample. And then, so in BioTuring, you can do these two analysis. For example, if you have the data come single cell, you can use BioTuring Pro to analyze this data, and then you can have the information from the single cell data, and then you can use SpatialX, which is the platform for analyzing spatial transcriptomic data to analyze like spatial data, including cosmic data here as well. But for the time limitation and so the purpose of for this seminar, so only the cosmic SMI data will be used on this webinar. So first, I would like to show we can get information from the cell type, including cell type proportion in different sample in the study. So in the study, they have like 3 different groups with healthy control and Crohn disease and ulcerative colitis. So one thing that in SpatialX that you can do is you can combine all of these data into one single data, and you can see here the condition we have different condition and labeling different sample with different um, like color here and with the metadata. And from that, in SpatialX, we are also able to do the cell type prediction, which is based on the single cell database from BioTuring talk-to data. From that, you can do, we have the option to do automatic cell type prediction, and you're also able to do the cell typing manually as well. So maybe I can show you quickly how to do that in SpatialX before we can move here, so we can know what we can do. So you can go to the SpatialX here, and then if you have any study, uh, you can go to the study tab here, and then you can go to combine data. For some here, I have like 4 individual sample, and from that I can combine the sample to on sample to do analysis at the same time. So I can select one of these sample, and then I just click combine. And after combination, you can see they will create another dataset we call on sample. From that, you can open to explore on a sample at the same time. And from SpatialX, we also have the option to do cell type notation based on the huge database from single cell data. So from that, you can go to cell type prediction, and the input that you need is Leuven clustering. And usually, when you submit the data from the platform like already do it for you. So from that, and then you can either choose the major cell type or subcell type. So major cell type is the overview of the big category, and then you can go to the subcell type here, and then the true major, and then you can also customize for the prediction. For some here, you can choose column at the sample, and then we can predict the cell using the single cell sample from the column. And from that, you can open the one not sure for, and then you can have the cell type information. So after you have the cell type information, one thing that you can do is we have you can go here and see they have some condition information. And from that, let me go here. So from that, to do the cell proportion, so you can go to the drag and grab here. And from that, you can go to cell proportion. The first thing you need to select is the condition, which is show different category, even from the healthy or disease sample. And then you can select the cell type information. And from that, you can construct a cell proportion as a project here. And you can also able to see the ratio of the cell at his map here as well. So that is how you can do with the SpatialX to able to confirm the raw data, confirm, and then you can combine data together to able to analyze, to do a cell type annotation, and then you can able to see the proportion of the cell in 3 different groups, for example. And let me go back here. So and then after you have the information from the cell type, and then you also combine the sample, what you can see here is when you see the 3 different groups. So one is the healthy control, and then you can have the Crohn disease and also the ulcerative colitis here. And one other thing that you can notice right away, they have like very different in cell proportion in terms of the major cell type here. For example, if I can hold over these myeloid cell, you can see in the healthy control, the myeloid cell is just around 6%, but in the ulcerative colitis is around 12%, almost like double the amount of that. And you can see also compare between different condition and different cell type. And from the information, you can see there an increase in the proportion of myeloid cell, and also in the B cell and plasma cell in the Crohn disease and ulcerative colitis compared to the control. And the one that I just showed you before is you can do cell type prediction at different level. You can do in a major cell type, you can go down to the subcell type, and for that subcell type, you can able to manually annotate into more subset of the cell type of your interest. And from that, I can get more detail about the cell type and the condition here as well. For example, I interested in the paper, the author interested in in a macrophage, which is expressed the NRG1. And you can see NRG1 macrophage is very different between different conditions. For example, in the healthy control, we just have the small proportion of the macrophage NRG1, and ulcerative colitis is a little bit increased. And then in the chronic disease, you can see the huge increase in this type of macrophage. And beside the seeing the proportion on SpatialX, you can able to see the distribution of the cell in the spatial with the spatial coordinates. For example, you can see in the healthy control, we can only see a few cells in the sample in the FOV region here. But in the Crohn disease, you can see there are lots of the macrophage in RG1 expanding in the Crohn disease. So suggesting that the macrophage may have some important function in the disease formation or in the disease progression. So, and then you can go beside like doing the cell proportion between three different conditions like healthy sample, Crohn disease, and ulcerative colitis. You're able to do the cell proportion compare between different sample confirm different patients to see the heterogeneity between different patients. And as very clear here, you can see very different in the proportion of cell between different patients, even though they are classified as the Crohn disease, for example. And for example, if I look into the macrophage and RG1, the one that I showed you before, and you can see if we just look into one cell type, you can also see the variability of the macrophage among different conditions. And this may contribute to the heterogeneity between the different patients when also involved in the response to different response to treatment condition. And beside doing the cell proportion, I also want to mention that in the SpatialX, we are also able to analyze gene expression and differential gene expression and also pathway analysis. But for the scope of this webinar, I more focus on the cell information, and for the gene expression analysis will be coming in the next SCSE Club. So next, so after we also know that there an expansion of the macrophage in the Crohn disease, and the next question that I would like to ask is any cell that may interaction with the macrophage for particular biological function in the Crohn disease. So to do that, I can in the SpatialX we have the option to do like neighborhood analysis. So when you run the neighborhood analysis, they will calculate the spatial correlation between different cell types. For example, here, because I'm only interested in the macrophage, because I can see the pattern of macrophage that increase or expanding in the Crohn disease. So I would like to interested in any cell that interact with macrophage, for example. So from this heat map, I can look to this heat map, and then I can see macrophage also have the correlation with the inflammatory fibroblast. And from that, we will click into this heat map, and you can see the expanding here, the position of the cell. Green is for macrophage, and then orange for inflammatory fibroblast. And you can see these two cells stay co-current in the similar area in the tissue, and suggesting that these cells may interact with each other for a particular function. And next is I would like to validate the neighborhood analysis by go down to the single cell level and then see any like particular region that really have this C cell that stay very close to each other. So to do that, I can based on the visualization on SpatialX, so I can go down to the single cell level, and then we can able to visualize the cell segmentation, so we can see where is the cell, and then also the cell battery as well. So you can see here the same, so green for macrophage, and then orange is for inflammatory fibroblast. And you can see these two cells stay in close proximity to each other. And if you focus on some particular area here, you can see the inflammatory fibroblast just stays next to the macrophage, and they can form the contact with each other, which is one of the information that we can validate that these two cells can interact with each other. So after the cell interact with each other, so the next question that we interested in to ask is about whether or not these cells communicate with each other and what is the pair of the ligand and receptor these two cells may potentially interact with each other. So to do that in the SpatialX, we have the option to do cell-cell communication. So which is gonna be released very soon. So from that cell-cell communication, you can enter the cell information, and then you can figure out what cell and which cell that may interact with each other, and then what is the potential receptor ligand that can facilitate that interaction. For example, here, if you go to cell-cell communication to analyze the interaction between the cell type that we've seen before, and if you look at the macrophage here and the inflammatory fibroblasts, which is the two cell type that we can see that stay very close proximity to each other in the neighborhood analysis and also in the single cell level. And then from that, we can see there are a high number of interaction occur between the two cell type. And when you click into this heat map, you're able to see the potential receptor or ligand that the two cells use for the interaction. And from that, we can go further for the desired experiment for wet lab for the validation between whether or not these two receptor ligand actually the one that the two cells use as the way to interact with each other. And we're also able to further analyze which pathway that may involve in this interaction. And beside the cell pair, we're also able to visualize the cell interaction network. For example, here is the whole cell interaction network between different cells in the tissue that we analyze. And then if you select one particular cell type, you can see which cell that cell type may interact. And the thickness of the arrow here is more number of interaction. So I can I will quickly go to the platform to show you how to do like neighborhood analysis and also cell interaction. So to do neighborhood analysis, so I can go here. So after you have the cell type information, which is come from maybe from your annotation or come from the prediction that we did before. So you have all the cell type information here. And then we can go to the option to analyze here. We have neighborhood analysis. And from that, you can enter the name for your record. And then we need to select the cell type information. And from that, you can run the neighborhood analysis. To save some time, I open the result I showed you before. So from here, you can see that we have the heat map showing what is potential interaction pair of cell. And from that neighborhood analysis, you can able to do, we call the spatial domain, which is identify different region in the tissue that have the one cell type or multiple cell type condensed in that region. So for example, here in the spatial domain 0, you can see here is more about inflammatory fibroblasts. And you can see the inflammatory fibroblasts is staying condensed in some area in the tissue here. Okay. And that is the neighborhood analysis. And then if you would like to do the cell-cell interaction and go down to the single cell, we have the option to do here as well. For example, here, this is the showing the cell center with different color represent different cell type. And to see the single cell level, you're going to go to the references here, go to cell, and then we select the cell segmentation here. Because in the Cosmic, we have the cell segmentation included here. So you can visualize that. And you can also able to customize the point size of the cell center and also the opacity of the segmentation. Yeah, you can visualize a different option. So from that, you can able to see which cell may potentially interact with cell based on this go down to single cell level. And from that, you can able to do the gene query here as well. So when you do the gene query, you're able to see first is the cell center gonna change the color. So the higher, the darker of color is the higher the expression. And we also able to have the option to show transcript here, which is able to show you individual transcript. So one single dot here represent for one transcript, and I can recite that here as well. So you can see like what cell express, for example here is AKT1, and which what cell like does not express AKT1 at the single cellular and single transcript level. Okay. And the last thing that I just show you in the end is the cell-cell communication, which is the new function that we have recently. So to do cell-cell communication, we also need the cell type information, which is can confirm the cell type prediction that I showed you before. And from that, we have the option to do the analysis here. We call the cell-cell communication. And one that you need is the input of the cell type information. And then you can run the cell-cell communication here. Okay. So to save time, I also open the one that I showed you before. So they have different options for you to view. So after the analysis is done, they will first show you the heat map of the sender and the receiver cell, and potentially number of interaction between these two cells. The darker color is the higher number of the interaction. And when you click into the square that contain the interaction between the two cells, you can see there are list of potential ligand receptor for that, and then you can view at the table here as well and download that potential ligand receptor. And at the same time, you can able to view the two cell on the view on the right here. And we also have the option to see how many interaction occur within the tissue. And also we also have the option to view as a network here. So you can see this is the whole interaction network in the sample. And if you'd like to focus on any particular cell for some macrophage, you can click here to the macrophage and you can visualize which cell that interact with macrophage or which cell the macrophage interact with, and go to an individual interaction here. Okay. Okay, so from that, let I go back to the slide here. So from that, I would like to summary the information that in this webinar here. So so from this study, we can able to see there are differences in the cell type proportion contribute to the patient heterogeneity in the IBD. You can see there are different patient group. They have different cell type proportion, and different individual patients they also have the very different in cell type proportion. The one that I just showed you is the macrophage NGO1, and then the expansion of macrophage NGO1 in the Crohn disease may contribute into the characteristic of the disease, and we could be further analyzed to understand more about disease. And the neighborhood analysis can show the cell the co-occurrence of the macrophage and the inflammatory fibroblast. But that's the one example that I show you here. But from that neighborhood analysis, you can get more information to explore about other cell type potentially interact in the close proximity together in the same tissue, in the same area of the tissue, and then you can further analyze, go to the cell-cell communication to analyze that. And single cell with cell segmentation validate the macrophage and the inflammatory fibroblasts to stay in a close proximity, and you can also see cells that make contact with each other, which is a very nice way to validate the whether or not these cells may potentially interact with each other. And last is cell-cell communication analysis, identify potential ligand receptor pair that macrophage and the inflammatory fibroblasts utilize for the interaction, which is a very nice tool for you to identify any potential ligand receptor to do further analysis. And from here, I think my colleague can put the poll for a list of questions and any suggestion that that you can have for the new feature that you would like to have in SpatialX. If you have any question about how to use SpatialX, so please feel free to drop your email in the chat box or in the enter into the question list, so we can able to help you further explore the spatial transcriptomic data. Okay. So while waiting for the poll, so I have some information about the resource. So all of the SCSE Club recording are available at BioTuring YouTube channel. So including this section as well. So if you go to the YouTube channel and you can type like at BioTuring, and you can go to a playlist. In that playlist, there will be a list called Single Cell and Spatial Exploration Club. From that, you can find all of the recording from the SCSE Club for in the past and up to the recent. So you can able to uh, watch again if you miss any episode. Okay. So for that, I can go to the question to see if we have any question. Okay, so okay, I think I have one question about cell. So beside the proportion that the cell that we can do to compare between different conditions, we also able to compare. We have the tool we do the annotation. So which is one of the tool that people interested in. So for example here, if we have this is one FOV from the cosmic sample, and then if the person that's interested in a particular region, for example, is they interested in this region, and they can use the lasso tool as shown here to annotate the region. And from that annotation, so you're able to see the pair cell proportion in this annotation, or you can able to do another thing we call like distance query. So you can query any cell, for example, surrounding like 100 micrometer around the region that you selected. So you can annotate that region, and then you can do the distance query. And then you can able to see the cell down here. Turn out this. So we can do the lasso tone here. Okay, yeah, some slow internet here. So for some here, so after do distance query, so they will label the cell that I just queried like 100 microns surrounding the region that I selected. And from that, I can annotate the region, like region of interest, and then 15 micron microphase around this RI. And from that, you can do further analysis based on that, the distance query. Okay. So, okay. Okay. So I get a question that how can do we create a cell condition as the metadata is not available on the original data? Yes. So yes. So thanks for that question. So for example, if we have on the sample here, we can have the option to do use the same thing like lasso tool to annotate cell. So for example, you can visualize on the cell here. Okay. Yes, loading here. It's a little bit of internet connection, so please bear with me. Okay, so we have all the data here. And for example, if we recognize this is, for example, is the disease, so you can lasso all of these cells and you can annotate. So basically, we just like select a population of cell and annotate that. For example, here you can do like condition. And then you can say that this is the Crohn disease, for example. Let I change this. Maybe I already have that. Okay. And then you can annotate that, and then you can continue to lasso and then append into that condition. So you can go next, and then you can lasso again. And you can annotate, and then you can go to the condition that you just established before, append to the condition, and then you can say this is the healthy control. Okay, so that's how you can create the metadata name condition from the data set if you don't have the condition metadata on that. Yep. So, okay. So let's see. Yeah, I think that is the question that I have. So so thank you very much everyone for joining the section. So I will drop one email here in the chat box. So if you have any additional questions, so please feel free to drop from the chat box and also from the email. So thank you very much for joining the section today, and looking forward for the new section, which is we can show you more about the SpatialX feature, including the one that I mentioned about this gene analysis. Yep. So thank you very much everyone for joining today. Have a good day.